Hola, buenos días a todos. Vamos un poco mal de tiempo, con lo cual vamos a empezar el curso de Patología Básica. Yo soy Juan Abraldes y primero de todo quería agradecer a la Junta Directiva por haber pensado en mí para organizar este curso de Patología Básica. Y agradecer también que ha sido un placer pertenecer al comité científico estos últimos 4 años. Entonces, voy a hacer una brevísima introducción porque vamos un poco mal de tiempo. El tema del curso es realizar una revisión de las herramientas actuales que tenemos para realizar investigación translacional, principalmente en pacientes. La primera charla hace una introducción de esto y después hay 2 charlas que son complementarias a la primera, es decir, que complementan cómo utilizar modelos animales para confirmar hipótesis o evaluar nuevos fármacos, nuevas líneas terapéuticas de datos que se han recogido en pacientes. Y, por último, una herramienta imprescindible para investigar las enfermedades, las hepatitis víricas, son los cultivos celulares de virus. Tenemos 3 excelentes speakers. El primero es Daniel Glichelou, él trabaja en París. Él es nefrólogo clínico, pero tiene una gran formación en ciencia básica y ha trabajado durante los últimos años en medicina translacional, principalmente en la clasificación molecular de enfermedades, en la predicción molecular de eventos, principalmente en el campo del trasplante renal. Dani, thank you for coming.
Thank you. So first I'd like to thank the organizer for this kind invitation, and I'd like to apologize for 3 things. The first is that I will speak in English. I'm sorry about that. The second one is that I'm only a nephrologist, so I'm very sorry about that, but you will have a few slides about renal transplantation also. And the third one is that the first anticipated speaker was my friend Alberto Sanchez, but he's getting married today, so it was impossible for him to attend the meeting. So I will try during the next few minutes to provide you a short overview of what could be the application of transcriptomic in the clinical side, and provide also a few basic insight, basic explanation regarding those techniques. So we will be in the omics world actually during the next few minutes. This is a central dogma of biology, from the DNA to RNA, messenger RNA, or non-coding but regulating RNA, protein and metabolite. And as you know, at each of these different steps, a number of techniques have been developed regarding genetics, transcriptomics, proteomics, metabolomics. We will just discuss today this application dealing with transcriptomic analysis of RNA in a given tissue or a given cell. So what can we learn from transcriptomics in clinical settings? The first thing we can get is to improve our understanding of gene and pathways involved in many biological processes. The second thing is that we may elucidate the function of a known gene, because generally we have a whole genome interrogation, and very often you found significant genes for whom you don't have any knowledge. And the third is that this kind of approach may also identify markers, biomarkers, for either diagnosis, prognosis, or prediction of disease or a response to a given drug, for example. So in clinical application, we may imagine that this kind of approach will help in classification, reclassification of disease, prognostication, individualization of treatments, and also discovery of new mechanisms and therapeutic options. So what are the methods to use in this kind of approach? You can quantify at a single gene level. And as you all know, the best method is quantitative PCR, and historically you use in your experiment Northern blotting, but this is not convenient for application in the clinic, or reporter gene-based, but definitely using transcriptomic studies, we need a quantitative real-time PCR. And what about the detection of multiple genes in given samples? There are also a number of methods. The newest one is this kind of high-throughput sequencing called massively parallel signature sequencing. We don't have time to discuss many aspects of this kind of approach, and we will focus today on microarray applications and other transcriptomic analysis in the translational medicine. So this is the basics of microarray. I'm sure you are familiar with it, but this is just a few words about that. You know that microarray are based on the hybridization of your cDNA sample in a glass in which have been spotted or synthesized in situ oligonucleotide from all the transcripts from a species. And this is a basic and first generation microarray that was organized as follows, with a glass, spot cDNA and spot oligonucleotide, and you could compare two samples, each of them labeled with a given fluorophore, and then compare the color of each spot, yellow, red, green. This is the first generation microarray with spot microarray and competitive hybridization between two different cDNA. But the most of the recent publication worked with this Affymetrix or Illumina platform, in which you have only one label RNA population per chip and then comparison between chips. This is what you will find in many of the papers dealing with transcriptomics. This is a heat map, and the heat map, as you know, is a graphical representation in which every column is actually a gene, and each row is a different sample. In this example, this is breast tumor, but it can be everything here. And you have a color code reflecting the level of expression in a given gene in a given sample. And in this example, you have here a cluster of upregulated genes in these samples. And usually you will find also a dendrogram. A dendrogram on the top of the panel and a side dendrogram. This dendrogram represents relation between two samples. This is a representation of what you can have as a dendrogram, and in a few words, a dendrogram is actually a representation of the clustering approach, and this clustering approach will group genes or samples into groups based on similarity and difference between them. Another critical point you will find in all the publication regarding transcriptomic and whole genome interrogation is this problem of multiple testing. As you know, multiple testing problems is the fact that when you multiplied two-side tests, you will find a number of false positives. And normally this is fine if you have only a limited number of comparisons. But in a microarray, you will do about more than 10,000 tests per experiment, and then you will find a huge number of false positive regarding your relevant genes. And this is the reason why in all the paper dealing with transcriptomics, you will find this characteristic, this point of false discovery rate, FDR. The best way to explain FDR is to provide you an example. If you have this example, more than 6,000 genes in your experiment, and your experiment found about 200 genes with differential expression using a p-value of 0.01. That means 0.01, that 1% of this number, 64 genes, would be expected to appear differentially expressed when they are actually not differentially expressed. With decreasing p-value, the false discovery rate also decreases, but so does the number of differentially expressed genes. And in that situation, the false discovery rate is 35%. 35 is actually the ratio between 64 and 184 here. So the lower the p-value, the lower the false discovery rate. And finally, what you have to do is to choose a p-value with balance, which balance number of differentially expressed gene and false discovery rate. We'll see an example a few slides later. So let's move to a few examples of what we can do with transcriptomics in clinical practice. As I said, I'm a nephrologist. I'm involved in transplantation, in renal transplantation. So during the next slide, I will show you an example of what we can do in renal transplantation, and thanks to Alberto's slide, a few words about liver transplantation also. In these different settings, we can use transcriptomics with different types of sample. You can use an invasive approach using the biopsy of the organ. You can use blood sampling. This is a minimally invasive approach with only a blood sample. And you can use, and particularly in renal transplantation, a non-invasive approach using urine from our transplant patients. So let's move to a few examples, and the first one I would like to discuss with you. You probably know this data more than myself. These are the results from Alberto Sanchez, and you remember that Alberto worked for many years on this kind of approach, trying to identify liver transplantation with operational tolerance. I mean patients who can withdraw all immunosuppressive drugs without any immunological complication in the long term. This is definitely a good model to study tolerance, because as you may know, operational tolerance is very high in liver transplantation. This is not the case in kidney transplantation, for example, but in liver, about 40% of patients will be able to stop their immunosuppressive regimen if they have been correctly identified. So this was a study from Alberto, and as you may know, patients with stable liver graft after 3 years of transplantation were included in this study with withdrawal in about 6 to 9 months and then 12 months follow-up. Some of them reject, but some of them didn't reject and were classified as a tolerant patient. At time of inclusion, a number of samples have been collected, including liver biopsy and also PBMC using a single blood sampling. And from these different samples, a number of transcriptomic analyses have been performed, including microarray, whole genome interrogation, and quantitative PCR assay. This is the first impressive result that has been showed. this is an heat map, as I showed you before, and this heat map showed that a number of genes were significantly differentially expressed in patients with tolerance and in patients with no tolerance. And this is the false discovery rate. And with a FDR of 0%, only 8 transcripts among the whole genome interrogation have been found to be differentially expressed in the two populations. If you put an FDR at 25%, you increase the number of significant genes up to more than 150 transcripts. The probably most important result from this analysis was quite unexpected result. This is the following. Among these significantly different genes, a number of them were classified in the functional of cellular iron ion homeostasis. This was unexpected, and this was actually confirmed by subsequent analysis. Among these important genes, one of them, AMP, which is epsilon antimicrobial peptide, was one of the most regulated between the two populations. And actually, in the blood from this patient, epsilon, the protein epsilon, was significantly correlated with the gene expression in the liver of this patient. And importantly, very simple tests, including epsilon quantification in the serum of a number of patients in different situations, actually confirmed that epsilon was lower in patients with non-tolerant at time of inclusion in the study than in patients who will become tolerant after drug withdrawal. An important thing, and as you all know, in all transcriptomic analysis we need not only a training set but also a testing or a validation set, and this has been done by Alberto, and he could confirm that a number of genetic classifiers perform very well in the training set as well as in the validation sets. So he also tried to test the same hypothesis but using a blood sample that could be more simple in clinical settings. And again, there was a significant signature of tolerance, but what he could find in this ROC analysis is that the best performance was for the transcriptomic classifier from the liver tissue, but actually blood signature also was highly significant and had a prognostic value. So this was the conclusion of this analysis, showing that intragraft transcriptional marker can be used to predict outcome of drug withdrawal in liver transplantation. So let's go to transplantation, but to kidney transplantation. I will show you very few examples, very few slides dealing with diagnosis of acute rejection using transcriptomic, prediction of acute rejection, prognosis of graft fibrosis. This is a study we did a few years ago, and in that study we tried to investigate microRNA signature within the graft, the kidney graft, during acute rejection, T-cell mediated rejection of the renal allograft. And what you can see in this heat map is that we could identify a microRNA signature of acute rejection, and it could lead to the discovery of new biomarker of rejection. In this rock curve, you can see also that a number of these microRNA had very nice AUC value of about 1, suggesting that this microRNA may be used as diagnostic biomarker of acute rejection in the kidney allograft. What about prediction of acute rejection? As I said before, in kidney transplantation we have access to urine samples, and Obviously, urine sample may provide huge information about what happens within the graft. And this is a study, these are unpublished results we obtained from a study that is still ongoing. In this panel, what you can see is the copy number of perforin messenger RNA in urinary cells of our transplant patient at 10 days post-transplantation, 1 month post-transplantation, and 3 months post-transplantation. You can see two groups here. Patients who will present rejection between 3 and 12 months, so afterwards, and patients who will not present any type of rejection between 3 and 12 months. What you can see here is that in the two groups, perforin was similar at 10 days post-transplantation. There was a trend at 1 month, but there was a significant difference at 3 months. And what you can see also is that patients with no rejection afterwards tend to decrease their perforin messenger RNA level in the urine, but patients who will reject between 3 and 12 months had an increased perforin messenger RNA level in their urine, suggesting that this kind of approach may also predict subsequent acute rejection 3 weeks or months later. And what about prognosis of graft fibrosis? This is one of the complications we have in renal transplantation. We have a number of patients who will develop fibrosis of their renal allograft, and this will lead to dysfunction of the graft and finally to graft loss. And we have a patient who will progress during the first year post-transplantation. The problem is at that step, at 12 months, you usually don't have any effect on the function, and then serum creatinine is not a useful tool to detect patient with fibrogenesis process. And you need a protocol biopsy, an invasive protocol biopsy, to detect this patient early in our setting. So we need a biomarker to detect that, and this has been done by a transcriptomic analysis in early protocol biopsy. This is the first study, a study in which a protocol biopsy at 3 months without any fibrosis. IFTA is actually interstitial fibrosis, and patients have been followed to 6 months, and at 6 months some of them presented fibrosis. So there could define two groups of patients, those with progression of fibrosis and those without progression. And if you test the transcriptome of the biopsy at 3 months, whereas all the biopsy are normal at that step, you can identify patients with a progression towards fibrosis. This has been confirmed in a very nice study published last year. In that situation, this is exactly the same design. Patient at time 0, 6 months, 24 months after transplantation, and they have protocol biopsy at each step and transcriptomic analysis at each step. You can see here two groups of patients, those with red dots with progression of fibrosis, those without progression in blue. And you can see here a number of gene sets providing information about T cell proliferation, B cell, NK cells, etc. And you can see that these two groups differ in their kinetics of these probe sets. And among these probe sets, one of them, dendritic cell migration at 6 months, was able to predict progression toward fibrosis at 24 months with a very nice predicting value. Let's go back to urine. What can we do in urine? This is a paper we published last year. We tried to identify a number of messenger RNA from urine samples that could be predictive diagnostic of fibrosis within the renal allograft. So we start from 114 samples of urine at time of biopsy and use a discovery set to define a gene signature and validate it in a validation set. And in a few words, we could identify a composite score using 4 different messenger RNA with a good diagnostic value for the probability of fibrosis within the graft using this totally non-invasive approach with a good AUC that we could validate in the validation set. So this is the last slide. As I said to you in the beginning of the story, we just spoke today about transcriptomic using microarray. There is a new approach which is next generation sequencing with a huge amount of data that are being to be produced. And you can see here the celerity, the kilobase per day per machine. And you can see that in the 90s we could also sequence few hundreds of bases per day. Today, this is the number. These are high throughput, very sensitive techniques, and these techniques may be useful in clinical practice because they become cheaper and cheaper today. So I thank you for your attention. I will be pleased to answer your question.
Thank you very much, Danny. I think we will leave the questions for the end of the session, so we will see whether we have time.